without worsening the second should be our goal to develop a novel TB vaccine. Or in other way around, there should be a delicate balance between the host immune response to control the infection, and also we need the tissue repair mechanism to limit the pathology. And definitely to achieve this criteria, we need a better understanding of our host defense to combat the infection. So our host defense comprises of two arms. One is host resistance, where the ultimate aim is to control the pathogen load. So the immune response associated with host resistance and the pathogen itself induce the tissue damage. And if the tissue damage don't get repaired, then the host will compromise with the survival. So we need another arm of host defense, that is disease tolerance. So disease tolerance reduce the negative impact of the infection by limiting the pathology. And the important point is it doesn't have any effect on the pathogen load. Unfortunately, we didn't pay a lot of attention to the disease tolerance and yeah, we didn't pay a lot of attention, but despite the fact that this area has been investigated and evolutionarily conserved and has been well studied and planned, there are recent reports demonstrating that the disease tolerance play an important role in different infection models. On the other side, the host resistance has a direct impact on the pathogen load. And that possibly the reason that we have gained a lot of attention in this area and has been constantly getting studied, and could possibly explain that why our classical approach of designing vaccines are more inclined toward the host resistance and mainly focused towards the T cells. So the classical concept of T cell mediated immunity was initially introduced in 1960 by Jacques Miller and Max Cooper. This group demonstrated that the thymocytes are involved in antigen recognition and also responsible for DTH response. This idea was further supported in 1973 by Robert North, and this group demonstrated that these thymocytes contribute to the containment of TB and it's independent of the humoral response. And one of the major evidence supporting that T cells are important in TB is from the host deficient in T cells. Either it's animal model where rag knockout mice or skid mice or HIV positive patients, they have reduced frequency of T cells, are super susceptible to TB infection. And one of the major criteria for vaccine designing is the memory capacity. And we know that when the antigen presenting cells get infected and they get encountered with the naive T cells, these T cells get primed, acquire the effector function, and with the pathogen clearance, these T cells acquire the memory response, either effector memory response or the central memory response, which can retain for a longer time period. And with having this criteria, virtually all our vaccines against TB are targeting the T cells. I mean, indeed, the T cells are important in TB, and there are ample of evidence to support that T cells are important in the containment of infection, but there's no evidence to support that T cells control the TB infection. So how ideal it is to design our vaccine, which are boosting the T cell response during TB. In fact, there are reports demonstrating that these uncontrolled activation of the T cells is detrimental to the host. So there's a study published by Joel and Sebastian group in Nature Genetics, and uh, this group demonstrated that unlike contrary to their hypothesis, that the human T cell epitope of MTB are hyperconserved. We also published our study in Science Immunology in 2018, and we demonstrated that the hyperactivated T cells is due to the deletion of mitochondrial protein cyclophilin D, which actually regulate their metabolic responses, render the mice more susceptible to infection, and that is due to the breakdown of disease tolerance. I'm just going to highlight some of the key data from this study. So when the PPF knockout mice, so the PPF gene is actually encoding for the cyclophilin D protein. So when PPF knockout mice were infected with MTB, so they have higher number of CD4 T cells in the lung at day 90 post-MTB infection compared to the wild type mice. And this was also shown with CD8 positive T cells. Despite having a higher activated T cells, I'm not showing data here, but they have and higher production of interferon gamma, they have higher proliferation. Despite their activated profiling, they do not have any differences in their bacterial load. These mice are having the similar load during the course of the infection. But when we look for the survival of these mice, the PPF knockout mice are more susceptible to TB compared to the wild type mice. Not only we, but there are other groups demonstrated and supporting the same concept. So there's another study published in 2010 by William Jacobs' group, and they showed that the PD1 deficient mice, so PD1 is an inhibitory receptor on T cells and actually control the proliferation and activation of the T cells. So these mice, upon exposure to the TB infection, have higher response to TB, but they're super susceptible to the infection. There's another report published in Science Translational Medicine, where the cancer patients, when treated with the PD1 blockade antibody, also have an incidence of tuberculosis. So this data possibly explain that why our current vaccines, our new vaccines targeting the T cell immediate immunity didn't show any success in animal model or clinical trial. I'm just quoting here two examples. One is from 2015, where they showed this MVA-85A vaccine didn't show any protective efficacy in that clinical trial. And another is in 2018, this clinical trial also didn't show any protective response in the end. But if the T cells are not the right candidate for designing vaccines, then what should be our approach? What should we need to target in the vaccine designing? So the question comes, what about innate immunity? And there are epidemiological evidence to support that innate immunity is involved in MTB elimination. So here, one of the data we have that uh, individuals from the hyperendemic area of TB, where they are at high risk of TB, are in fact are TST negative, but they are capable to control the TB infection. They are naturally resistant to TB. There's another report from the household contact of active TB patients. So 30 to 50% of the household contact of active TB patients are TST negative, and they're also resistant to TB infection. Having this idea, I'm just going to brief all of the whole idea from the figure that I have adapted from a review article written by Maz and Marcel, that when the person get infected with the TB, the innate immune response is activated. So these innate cells phagocytose the pathogen and then control the MTB infection. And then this whole phase of the infection, the early phase, contribute toward the host resistance. But if the innate immunity fail to provide protection, then the T cells get activated and initiate the formation of granuloma, and that is come under chronic phase of the infection. And then these T cells are required for the containment of infection and contribute to the disease tolerance. So this sounds really great that we have an innate arm which actually are responsible for controlling the MTB infection. But since we are talking about the vaccine designing, and we need the memory response as one of the major criteria. So does innate immunity have memory capacity? So we know that the 95% of the species on the earth have innate memory, completely rely on innate memory. And it is very unlikely that this memory trait is just confined to the adaptive immunity, which is actually evolved in the vertebrate. So we know that there are evidence that the plant are capable to provide protection against subsequent reinfection through a mechanism. One of the mechanisms is systemic acquired resistance. Not only plant, but invertebrate are also known, have been shown to provide protection against subsequent reinfection. And in fact, there's a data from the vertebrates. When the humans who get vaccinated with different vaccines, they have some kind of protection against non-specific infectious diseases. With all these data and the recent concept, which was boosted by Dr. Mihai Mithia, support that when the monocytes and macrophages get infected with the pathogen or with the vaccines, then these innate immune cells get epigenetically reprogrammed and acquire the activated function. And these cells are more efficient in controlling the subsequent infection. This whole concept is defined now as trained innate immunity. So again, at least we have one major criteria fulfilled, that innate immunity have memory capacity. But we always thinking in terms of long-term memory response. But the short lifespan of innate immunity is limiting the efficacy of innate response to be considered in vaccine designing. But this can be overcome because we know that the hematopoietic stem cells, which are actually produce millions of immune cells every single day, including monocytes, macrophages, red blood cells, and platelets, under the process known as hematopoiesis. And this is a complex process, required sequential events to give rise to the mature immune cells from the hematopoietic stem cells. I'm going to briefly summarize those steps, that how these hierarchy look like. So we have on the top long-term hematopoietic stem cells. So these are the long-lived cells, have the maximum regenerative capacity. And when there's a demand of the immune cells, then these cells give rise to the short-term HSCs, which are actually transient hematopoietic stem cells, and further give rise to the MPPs, these are multipotent progenitor cells. These multipotent progenitor cells give rise to either myeloid progenitor cells or can also give rise to the lymphoid progenitor cells, termed CLPs, and further give rise to the T cells, B cells, and NK cells. However, CMPs can either give rise to the granulocyte monocyte progenitors or megakaryocyte erythrocyte progenitors. So having this nice concept in front of us that hematopoietic stem cells can produce immune cells, and these are the long-lived cells. So in our lab, we demonstrated that the excess of BCG in the bone marrow reprogram these stem cells and generate protective train immunity. So when the BCG get access to the bone marrow, these hematopoietic stem cells get reprogrammed, and they acquired a unique transcriptomic profiling and also acquired some epigenetic changes. And these epigenetic changes and the transcriptomic signature get transmitted to the downstream progenitors and could reach up to the monocytes. And these monocytes are highly efficient in controlling the pathogen or in the subsequent infection. So this whole idea demonstrates that this BCG reprogramming generate trained immunity, and this whole process is type 2 interferon dependent. So here we demonstrated one of the possible mechanisms that how BCG reprograms stem cells and provide protection against pulmonary infection. But this overall idea of systemic administration of BCG is not very new or novel here. In fact, the superiority of this approach, the BCG vaccination administration intravenously or systemically, have been shown superiority even 1970s. So I'm just quoting some of the studies here which published in 1970 and 1972, and they demonstrated that BCG vaccination upon intravenous injection have a superior effect on providing the protection against pulmonary MTB infection. And this another report published in 2016, they also showed the superiority of BCG against MTB infection. There's a recent paper published by Robert Cedar in 2020 in Nature, and they came up with a very interesting data that intravenous BCG immunization prevent tuberculosis in macaques. So this gave a very good concept that systemic BCG administration has a unique effect on the immune response and protect the non-human primates or prevent the tuberculosis infection in the macaques. And the interesting fact is that this whole concept of reprogramming of stem cells is not just confined to the live microbes. Even the adjuvant or microbial ligand can induce a trained immunity. I'm just showing here data from our recent study that we published with Mihai Nitya, that this beta-glucan, which is a naturally occurring polysaccharide and found in the cell wall of fungus, including yeast, reprogram the stem cells and promote myelopoiesis. And these myelopoiesis give rise to a trained monocytes and macrophages, which are highly efficient in controlling the MTB infection. And I'm just showing data here from that study that when the mice were treated with beta-glucan intraperitoneally, day 3 and day 7 prior to the MTB infection, and when we looked for their survival, So the blue line is for the mice receiving the beta glucan prior to the pulmonary infection. They have a superior, very high survival compared to the wild type mice not receiving the beta glucan. And this whole survival or the beta glucan effect on the protection is IL-1 dependent. When we have IL-1 receptor knockout mice treated with a beta glucan, and when we look for their survival, there's no difference in the survival of the mice. Either they receive the beta glucan or they do not receive the beta glucan. So with this overall idea that the reprogramming of stem cells generate trained innate immunity, the next question that we have, and we were very curious to know, what impact does the MTB have on HSCs, and what consequences does it have on trained immunity? And there's another angle to look for the MTB on HSCs, because the suppression of bone marrow in TB has been documented even 100 years ago. And there are few reports indicating the MTB hide in the mesenchymal stem cells, which is also one of the kind of stem cells found in the bone marrow. So having this idea, we started our study, and the first experimental setup we have is pretty much similar that we have in our BCG study. So the idea is to do the side-by-side comparison of BCG vaccination and MTB infection and looking for their effect on the hematopoiesis. So here we have two groups. As I mentioned, we have a similar approach that we did before. The mice were vaccinated with BCG, and we vaccinated them intravenously with 1 × 10⁻⁶ bacteria. The other group is infected with MTB IV and have received the similar number of bacteria. And then we looked for the phenotyping of these mice in the bone marrow to look at long-term HSCs, short-term and MPPs, including the myeloid and the lymphoid progenitors at different time point of the infection. Here is the data that showed this MTB IV infected mice are super susceptible to TB infection. They all died by day 120 post MTB infection, and their mortality started after day 40 or day 45 post infection. So when we started looking for the phenotyping in their bone marrow, the first population we have here is LKS. I completely forgot to mention that the top 3 population on the hierarchy of these hematopoiesis, long-term HSCs, short-term HSCs, and MPPs, we collectively call them together as LKS population. So this is the population representing these blue long-term, short-term, and the MPPs on the top. So what we observed here is that at day 28, irrespective of the BCG or MTB infection, both the mice have similar level of LKS expansion. But one thing is striking, that MTB infected mice have started earlier proliferation of LKS compared to the BCG vaccinated mice. And one thing which is also striking is that at day 90 post-vaccination of BCG, everything is coming back to the baseline level. However, we do not have this time point for MTB infection because, as I mentioned earlier, that our mice started dying after day 45, so we didn't include this time point in our phenotyping. Having this idea of LKS expansion, we started looking for the downstream of this population to assess the myeloid and lymphoid progenitors. So here's the data for CMP, which is common myeloid progenitors. So here, if we look for the BCG effect of BCG vaccination on CMP, we could see that there is a decline in the number by day 14 post-vaccination, but then later on, these numbers get recovered and come back to pretty much similar level by day 90 post-vaccination. Contradict to that, the mice infected with MTB, they show a higher decline, a reduction in the frequency of CMP, and these numbers are continuously lower till day 28 post-MTB infection. And the similar trend we observed with the GMPs, that these GMPs started declining slightly, but they recovered back, but the trend of going down was consistent in the MTB IV infected mice. So contradict to the myeloid progenitors, when we look for the lymphoid progenitors, we observed that these MTB infected mice have more number of CLPs compared to the BCG vaccinated mice. So this is very striking that a BCG and MTB have a distinct profiling of these myelopoiesis and lymphopoiesis, but we always have a question that from the pathogen side, what is responsible for these differences in the CMP, GMPs, or CLPs. So to address this part, we collaborated with Dr. Marcel Baer, and because we know that the BCG and MTB have one classical difference is the deletion of RD1 locus. So we used these RD1 mutant strain of H37RV, and we infected our mice with that strain along with MTB IV and vaccinated with BCG IV. And we didn't observe any difference in the LKS population at day 20 post-vaccination or infection. But this is pretty much similar what we have observed with BCG, MTB in my earlier data. But when we looked for the downstream progenitors from the LKS, so here the x-axis is indicating for the mutant strain of H37RV, that is Delta RD1 strain, and the other y-axis is representing the BCG. And what we could see that both the strains have exactly inclined on the same line. So they have a pretty much similar trend of the downstream progenitors as the BCG. There's no difference in the BCG and H37RV. So this data indicating that MTB suppress myelopoiesis and promote lymphopoiesis, and this whole differences are RD1 dependent. So next we want to move forward with our functional data, that how these changes in the CMP, GMP are correlating with our functional data. So we assess the functional capacity of these macrophages to control the MTB infection. And to address this part, we set up our experiment where all the groups, one group was vaccinated with BCG, infected with MTB or S30 Delta RD1 strain of MTB. And after 4 weeks of establishing the infection, we drive the macrophages from these mice. But one thing that I would like to mention here, that all these macrophages were cultured in presence of TB drugs, just to get rid of any in vivo bacteria in these macrophages. And then these macrophages were harvested and then infected with in vitro to assess the protective capacity by a CFU assay. So here's the data, and I'm going to emphasize at day 5 post-infection. So the black bar are for the BMDM derived from the BCG vaccinated mice. And we can see here that they have a better protective capacity to control the MTB infection. And this is pretty much similar to what we have observed in our earlier study. And the Delta RD1 is also showing pretty much a similar trend, that they are better in controlling the MTB growth. Unlike Delta RD1 strain or BCG, BMDM, the macrophages derived from the MTB infected mice are more pervasive to the MTB growth. So this whole idea suggests that MTB suppress myelopoiesis and impair train immunity, as we could see that macrophages are not efficient in controlling the MTB growth. With this data, we want to move forward to look for the transcriptomic profiling of these HSCs and the progenitors. And to address this part, we did a collaboration with Dr. Luis Ferrero from Chicago. But I would like to mention here that when we were setting up this experiment, I and Maz, especially Maz, were so confident that we will end up with some unique signatures with BCG and some unique signatures with MTB, which will be really helpful, and we will have some kind of dichotomy to address this part. But we ended up with this figure. So here we have a gene regulation in the BCG or MTB HSCs and MPPs, and this dense blue area indicate that they're completely overlapping with each other. And you can imagine that how disappointing is this to look at this figure, but we are thankful to Louis' group. They come up with some very exciting data. So when they looked for some of the pathways, what they observed that there are genes involved in some of the pathway which are highly enriched, either in response to the MTB infection or in response to the BCG vaccination. So on top of that, we have interferon alpha response. So these genes are highly enriched in MTB infected mice. And another pathway which we observed having a highly enriched gene for BCG vaccination is heme metabolism. So these two we want to pursue with these, we thought to pursue with these two pathways to continue to find out our mechanism that how BCG and MTB behaving differently. But our first attempt was to establish our observation in pulmonary MTB infection. So we infected our mice with MTB through aerosol route with the deposition of 50 to 100 CFU in their lung. And following the same trend, we started looking for the HSCs and MPPs during the course of the infection. But we were also interested to see what is the kinetics of getting MTB into the bone marrow. So what we observed that MTB was getting access to the bone marrow as early as day 10 post-MTB infection. And these numbers were continuously rising, as we could see by day 120 post-MTB infection. And this increase in the bacterial burden was very well correlated with the expansion of LKS. So we have an increase in the number of LKS as well. And when we look for the downstream of CMP, GMP, and CLP, we observed the same trend, that is a decline in the CMPs, and these numbers were significant by day 120 post-MTB infection. And we also observed the same trend for GMP. Contrary to the CMP, GMP, there was an increased trend in the CLPs. And this data is pretty much following the same trend of what we have observed with our IV infection model. So it suggests that MTB reprogram these stem cells, suppress myelopoiesis, and promote lymphopoiesis. But again, in this pulmonary MTB infection model, we want to correlate these differences in functional readout. So what we did, we derived the macrophages from the mice, which were at day 120 post-MTB infection. And again, we follow the same trend, same experimental protocol as I have discussed before. And we looked for their protective capacity for in vitro MTB infection. So here we have data from the macrophages derived from these aerosol infected mice. And these macrophages are inefficient in controlling the MTB growth, or we can say they are more permissive to the MTB growth. So we have an idea that these macrophages are not controlling, and further we supported in our pulmonary model that MTB suppress myelopoiesis and generating impaired trained immunity against TB infection. We also looked for the single cell analysis of these HSCs and the progenitors, and we ended up with so many clusters within one population. As I mentioned here, that short-term HSCs have different clusters, MPPs have different clusters. But one of the things which was unique here, that all the clusters have an imprinting of interferon response. So from our data from our bulk RNA-seq, which also shows an upregulation of the gene interferon alpha response, and also from our single cell RNA-seq data that they have a signature of interferon alpha from all the population, assuming that the signatures are getting imprinted on long-term HSCs and getting transmitted to the progenitors. So we took this information and thought to look for the mechanistic approach. And also there's a very supportive literature available that type 1 interferon is detrimental to the host in MTB infection. So we started looking for type 1 interferon pathway. So first thing we did is we infected our wild type and IFNAR knockout mice. So IFNAR knockout mice don't show any type 1 interferon signaling. So when we infected them with intravenous MTB, these IFNAR knockout mice are more resistant to the TB infection. And then we started looking for the bone marrow phenotyping. And here I'm showing data for the myeloid progenitors and lymphoid progenitors. So one thing which is very interesting is that these IFNAR knockout mice are following the same trend as the wild type mice. They have a huge reduction in the frequency or in the absolute count here for day 7. But at later time point, these mice are capable to recover these numbers. So they have more CMPs by day 28, more GMPs. However, we didn't observe any difference in the CLPs, considering that this suppression of myelopoiesis is type 1 interferon dependent, but possibly there's some other mechanism that is regulating the lymphopoiesis in MTB. And again, we want to confirm these changes in the myeloid progenitors in terms of functional data. And we drive the macrophages from these different mice, the wild type infected mice and IFNAR knockout mice having their respective control. And what we observed earlier, that the wild type macrophages derived from the MTB infected wild type mice are more permissive to the MTB growth. However, there's no difference in the BMDM derived from the IFNAR knockout mice, irrespective that they're infected or they're not infected, suggesting that these MTB suppressed myelopoiesis and this impaired trained immunity is type 1 interferon dependent. But still the question still remains that what is leading to the suppression of myelopoiesis? What is happening to the CMPs and GMPs during the course of the infection? So when we look back into our RNA-seq data, we observed the increase in the gene expression of RIPK3 in MTB in the HSCs of MTB infected mice compared to the BCG or to the control mice. And there's a literature available, and we also have demonstrated that MTB infected macrophages undergo necrosis, and it is through BCL-XL or RIPK3 dependent manner. There are many other studies available. I'm just quoting here. There's another study which showed that type 1 interferon drive this cell death in the hematopoietic stem cells and is also RIPK dependent. So having some idea from our RNA-seq data and also having support from our literature, we look for the necrosis in the CMPs from these different treatment. And what we observed that the CMPs from the MTB infected mice have higher frequency of necrotic cells compared to the BCG vaccinated or the control mice. And the same we observed with the GMPs. I'm not showing here data for CLPs, but there's no difference observed in the CLPs in those mice. And to further confirm the role of RIPK3 in the necrosis of these cells, we infected our RIPK3 knockout mice with MTB and also did the phenotyping to assess the CMPs. And we observed that the RIPK3 knockout mice have more number of CMPs compared to the wild type mice. And the similar observation we have with the GMPs, and there's no difference in the CLPs. So these cells are undergoing for more necrosis, and that is the reason we have suppression in the myelopoiesis. But still the question is how type 1 interferon is inducing the necrosis in these myeloid progenitors. So I'm just going to recall our RNA-seq data. As I mentioned earlier, that another pathway that was having a highly enriched gene in BCG vaccinated group is heme metabolism. And one of the micronutrient associated with heme metabolism is iron. 70% of the iron is a part of the heme. And we know it's a vital micronutrient, and it's involved in the physiological function of the cells, include from respiration to the DNA replication. And when we look particularly to understand the hematopoiesis, there's a highly enriched literature available to support that iron is critical in maintaining the homeostasis of hematopoietic stem cells. And in fact, there's a different way that has been explored to address that iron is important in hematopoiesis. So in this study, they used a model having a transferrin receptor deficiency and showed that the iron uptake play an important role in hematopoiesis. In fact, there's a paper supporting that type 1 interferon also dysregulates iron metabolism or iron homeostasis and enhances the susceptibility to the fungal infection. So taking all these into consideration, we started our study with transferrin receptor. This is one of the molecules which is involved in iron uptake and also has been shown for hematopoiesis. And the important thing is it's easy to follow up as well. We have to do flow cytometry with this molecule. So I'm just showing here data for with the histogram. So these open graph are from the CMPs for the MTB IV infected mice, and it is clearly we can see that there's a high expression of CD71, which can be easily appreciated by these bar graphs. So the expression of CD71 in terms of MFI is very high in MTB infected mice compared to the BCG vaccinated or PBS control mice. To further confirm that this expression of CD71 is type 1 interferon dependent or not, so we use the same approach. We phenotype these mice, the wild type infected with MTB or if not infected with MTB, and we look for the CD71 expression. And again, there's differences we can appreciate in this bar graph that is, if non-knockout mice infected with MTB have reduced expression of CD71 compared to the wild-type infected mice. Further, to link this whole process to the cell death, we looked for the necrosis of these CMPs in both genotype, wild-type, and IFNAR, and we can appreciate here that these IFNAR knockout mice have lesser frequency of necrotic cells post-MTB infection compared to the wild-type mice. So this gave an indication that RN is the type 1 interferon is regulating the RN metabolism and inducing the necrosis of the CMPs and GMPs in this model. And we know that RN is, as I mentioned, RN is critical for physiological function of the cells, but at the same time is important for the growth of the pathogen. And that is possibly the reason that this whole RN metabolism is tightly regulated. I'm just going to briefly explain here that this iron is associated with the transferrin receptor. So if the iron is available in the free form, it has the tendency to leads to a negative impact. So in most of the cases, the iron is available in conjugated to the protein, so it can stay in the inert form. So the iron transferrin get transported through the transferrin receptor in the endosome, and then it get released from the DMT1 channel. And then this iron is either mobilized to the mitochondria for the heme synthesis and performing the other function, or in the other way around, it gets stored in the ferritin protein. So ferritin is a protein that stores the reservoir of the iron. And the other thing about the iron is that it exists in two forms, oxidized form and the reduced form. So here, when it is in the conjugation with the protein, it is always in the inert form. And when it gets free from those protein, it acquires the +2 form, and which has a tendency to generate the free radicals and also induces the cell death. So we have one approach that if we have a deficiency of ferritin, then this iron is available in the +2 form and has a major tendency to generate free radicals. I'm not going to explain the chemistry here because I'm very bad at that. And that leads to the cell death of the immune cells. So to confirm our all observation that this iron metabolism is important in hematopoiesis, and then this dysregulation could lead to the suppression of myelopoiesis and have an impact on the trained immunity, we did the collaboration with Dr. Miguel Suarez from Portugal, and he provided these tamoxifen-inducible Cre FTH lox mice. So when we give tamoxifen to these mice, then this Cre recombinase activity leads to the deletion of FTH from these mice, and they become complete knockout for ferritin. I mentioned that ferritin is a protein that stores the excess form of iron. But the limitation of this model is that once the ferritin get deleted, these mice died within 7 days due to thermodysregulation. So what we did, we utilized these mice for making a chimeric mice. We took the bone marrow cells of these mice and adaptively transferred them into irradiated congenic CD45.1 mice. And after 8 weeks of reconstitution, we gave the tamoxifen to these mice. And after 1 week of wash period, we confirmed that there is a deletion of tamoxifen in the hematopoietic compartment. And also we did the analysis of bone marrow or infected them to examine their susceptibility to TB. So what we observe here that there's a decrease in the frequency of CMPs in these ferritin deficient bone marrow compartment in these mice. And also there's an increase in the CLPs in these mice. So this is pretty much supporting what we have seen earlier in our MTB infected model. And when we drive the macrophages from these ferritin deficient mice, we observed that these macrophages derived from these ferritin deficient in the bone marrow compartment are more permissive to the MTB growth. And as I mentioned, that one group of our experiment was infected with the MTB through aerosol route to look for their survival and the other parameters. What we observed here is this green line is indicating the mice having deficiency in the ferritin in their bone marrow compartment and get infected with TB. So these mice are super susceptible and they all died before 50 days post-MTB infection. There's a second group which is having a deficiency in the ferritin compartment in their bone marrow, but they do not get infected. They were not infected with TB, but they also have a higher mortality compared to the mice which have intact ferritin compartment in their bone marrow, suggesting that these mice get more susceptible to TB infection, but the overall, this iron dysregulation have an impact on the survival of these mice. When we look for the bacterial burden in these mice, we observe that there's an increase in the bacterial burden in the mice having deficiency in the ferritin compartment compared to the mice having a ferritin intact in their bone marrow. So I'm just going to summarize this whole study here, that this excess of BCG in the bone marrow reprogram these stem cells, promote myelopoiesis, and induce trained monocytes and macrophages, which are protective in nature. And this whole process is type 2 interferon dependent. Contradict to that, when MTB get access to the bone marrow, it reprograms stem cells, but suppress myelopoiesis. And this myelopoiesis suppression is dependent on RIPK3 and iron dysregulation. And the monocytes and macrophages derived from these CMPs are super susceptible to infection. And this whole process is type 1 interferon dependent. And in the end, I would like to thank my Maz for sure. He gave me an opportunity and constant support and guidance, including my lab member, especially Jeff, who is a co-author on this paper. And we worked together. Like it was really encouraging to work with him and my all other lab members, collaborators. And thanks to the BSL-3 facility, who really made this project going on very smoothly, Histopathology Core, and my funding agencies. Thank you. Happy to take this question.
Okay, thanks, Nargis. That's a very, very impressive amount of work.
Thank you.
Most of the references were for the cell paper. So there must be a huge amount of data in one paper, my goodness. Anyway, congratulations, really excellent work. I just kind of trying to bring it back. I'm still always amazed how BCG seems to have been a fairly good vaccine despite everything. 1923, and a rather simple method of development. But I wonder whether now you have thoughts vis-a-vis, sort of where do we go in terms of human vaccine development, and what would be kind of the next step if we were to look towards, what should a vaccine look like, effective vaccine for TB?
So I think we should work a little bit more to find out what are the BCG component possibly is involved in the trained immunity, because there are papers supporting that BCG trained immunity probably work to the NOD2 ligand. So if we have any component of the BCG, and that component, if we can target along with our other component of the vaccine approach, possibly could help in designing or improving the vaccine. But we don't know what is that essential component of the BCG. But I think we need to investigate that which component or which signaling pathway is important in inducing trained immunity in the BCG. That's what my thought is.
Great. Okay, Marcel has a question. Marcel, do you want to go ahead?
Yeah. Hi, Nargis. I guess the first question is, that photograph of a super spreading event, did that happen after 8 p.m. or was that before the curfew?
It's before curfew. It's before curfew. It's like a dream for me, and I want to have this.
Okay, good. Just want to make sure that Maz is following all the guidelines. Towards the end of the presentation, I think I can see now that it's on slide 29. You showed us that the FTH knockout mice infected with Mycobacterium tuberculosis died in, I think, a median of about 30 days.
Right.
Yeah. Did they die of pulmonary tuberculosis, or did they die of a systemic illness with bone marrow dysfunction and something else non-pulmonary as their cause of mortality? Do you know?
So we did the phenotyping of these mice, and definitely there is a defect in their bone marrow compartment, and we could see that there's a suppression of myelopoiesis and so on. But at the same time, they also have a higher bacterial load in the lung, and we could see an increase in the spleen and the bone marrow as well. And we didn't observe any other like thermodysregulation as has been reported before. So we are assuming that they died because of pulmonary MTB infection. Because the other group who didn't have the MTB infection, they died, but they died because of the defect in their bone marrow compartment in the iron dysregulation.
So to be clear, if you took an H&E of their lung,
Okay.
they ran out of breathing space?
I don't have the lungs histopathology on these mice. I'm sorry, I could not answer that question.
Yeah, because as Dick and some of the other clinicians know, when you get mycobacteria in the bone marrow,
Right.
then you have something that's more similar to disseminated M. avium of an AIDS patient.
Okay.
Where people died of disseminated M. avium, but they weren't dying of pulmonary disease.
Okay. Okay.
So that's why I wasn't sure if your higher CFU means they're dying of, because your animal model is partly a lung disease model and it's partly a systemic model, which is one of the great challenges of TB in general, right?
Right.
People lose weight and people lose their appetite, and to understand which is compartmentalized and which is systemic, you have potentially a great tool to study that depending on what your cause of death is.
Right. For sure. I don't have the histopathology on these mice, so definitely that's important to look for in the next experiment.
Thank you, Nargis.
Madhu, please go ahead.
Nargis, amazing work. Kudos, and I hope you do exceedingly well wherever you end up going. Brilliant work. I just counted, there are 25 randomized trials registered or ongoing on BCG for COVID-19, okay? It's absolute bonkers. 25 trials, multiples of millions of dollars already invested in BCG for COVID.
Right.
And what do we have for TB? The GSK vaccine candidate is about to enter phase 3 in 2024, according to the Gates Foundation, with the results available if it is successful in 2028.
Right.
That is the abysmal situation of the TB vaccine pipeline. And it just frustrates the hell out of me as to we can't even get a good candidate into phase 3.
Right.
And we have no guarantees that it's even going to work, right? That's where we are with the vaccine candidates for TB. But I'm told that we will have the results of the BCG revaccination trial earlier than that. What is your take on BCG revaccination, especially if you're just going to use the same route as we've been using all these years? There is really no new pathway for the vaccine. Do you see any hope in revaccination?
I mean, if I remember correct, the clinical trial that I have shown before is they have the neonatal BCG vaccination and following up the BCG revaccination. But to my understanding, they didn't achieve a protection level that we need for the vaccine. So based on that, I can say it's still not very clear that the BCG revaccination could be an ideal approach to deal with, but might be changing the route of vaccination might have some impact on that.
Thank you.
Any other questions? Erwin, you must have a good thought-provoking question, I'm sure.
You're on mute, Erwin.
No, he's pretending. He's pretending he's on mute. Go ahead, Erwin. Are you truly, you can't say anything, Erwin? Sorry, okay.
Nargis, can I ask you a question?
Sure.
So what would be your expectation in this FDH model if you will vaccinate them with the BCG?
In fact, if I remember, we did those experiments that we vaccinated these mice with BCG vaccination, and they still have a defect in their bone marrow compartment. We have not achieved to that level of the myelopoiesis as we have compared that with the wild type.
No, I understand, but in terms of survival and control of even vaccination, do you have any data on that? Or?
Not on the control of infection, but it seems that iron is important in trained immunity. So might have some impact, but I don't have data for that side.
Because your transcriptomic data show that there was a shift in the BCG group, if I remember correctly.
Right.
And that
Yeah.
was the indication that we went after this pathway, but I guess we haven't done a careful study on BCG side.
No, we have not done that part. That's true. That's true. In fact, I'm trying to look for the literature that, because it's a report says that both all mycobacteria required iron, but I was curious to know, like, what about this iron is playing such an important and distinct role, then how BCG is extracting the iron from the host, like what could be the possible mechanism? So definitely there could be a different strategies utilized by BCG and MTB and its impact on the immune response, for sure.
Thank you.
Any other thoughts, issues, questions from the assorted throng? Anyway, very interesting talk, Nargis. Really, again, congrats.
So sorry, may I ask maybe the clinician, I'm wondering what is the frequency of bone marrow failure in active TB patient, and how common it is? Because I know that you don't X-ray the bone marrow, so I don't know that.
Sure.
If we carefully look at this and what is the information.
So in people, so this kind of refers back to what Marcel was talking about a little bit, which is that people with pulmonary TB that don't have, disseminated disease may have mild anemia, sort of the anemia of chronic disease where there's suppression, but they often have thrombocytosis and they may or may not have leukocytosis. So there isn't much evidence of a general marrow suppression. People with disseminated TB, so miliary tuberculosis, they certainly can have, then their marrow can be packed with granulomas and TB, and then they will have a diffuse pancytopenia, or can have. So
what is the frequency of those people?
So that's 1 or 2% really. And it's really not, so it's not typical of pulmonary disease, which is, 80% of patients.
Yeah. Maz, I think about 15 years ago, I reported a case where the miliary TB was so extensive that the patient phenotypically had AIDS and had HIV negative cryptococcal meningitis. So we had to treat the cryptococcal meningitis, and while treating the miliary TB, the CD4 came up, and then just simply by treating the TB, we could stop the cryptococcal treatment. So that was clearly a bone marrow failure, but it's also clearly reportable as a case report because it's not the norm. It's not what we see typically in the TB clinic. It's an outlier.
So do we know that people who die of TB, they die of really pulmonary respiratory dysfunction, or they die because the TB is disseminated to different organ and cause the death? I mean, because going back to Marcel's point, I'm wondering, is that their entire lung is packed with the bacteria and inflammation, that there is no air sac to breathe, and that's why they die? Or, some part of the lung is still functional and the bacteria is gone somewhere else and that caused the death?
So, I'm not sure that I've really seen a clear-cut autopsy series where people, looked at the bone marrow and at the lungs. I think most patients with pulmonary TB, the assumption at least is that it's a respiratory death. Honestly, it's not that common, so I don't think many of us clinicians have actually seen that much. They also, of course, patients die of complications, so they develop a pneumonia, pulmonary embolus, or hemoptysis, and that's actually the kind of the moment, the ultimate cause of death, if you will, is a secondary complication. Myocardial infarction, people who are older, that sort of thing. But I've not seen there must be autopsy literature on how often the bone marrow is affected. I just haven't seen it. I don't know, maybe Marcel or Madhu, you've looked at these.
No, certainly in the NTM people I've seen with relatively mild immune suppression who get disseminated MA via Amgen or whatever, then they tend to have overwhelming bone marrow disease. My understanding is most people who die of pulmonary TB exsanguinate when the granuloma erodes through an arterial or has some other local problem like Dick says. And for that reason, I think the mouse model is in that way a bit imperfect because usually the mice who die of TB are suffocating because 95% of their airway is destroyed by the disease. While I think that the human who dies of TB does not need to have 95% destroyed. It may only be 20% destroyed, but if in that 20% you have a fatal event, then that will do you in. So that to me is probably one of the key differences between the mouse and the human cause of death.
Fantastic, thank you.
Can I also follow up on Matt's question? I have come across with some TB reports as well that there is bone marrow fibrosis, but still the question is same, that what frequency is there for the bone marrow fibrosis in TB patients?
I'm not aware if anybody's done that, but Nargis, is Faz on the line?
I don't think so.
Yeah, because Faz and others are starting to look more and more at the long-term complications of TB. And I think there has been a risk that we kind of say, your 6 months are done, congratulations, here's a certificate and prosper. And maybe these are the kinds of things that could be built into longitudinal cohorts where you take people who are diagnosed, treated, and cured of TB and you follow them for 5 to 10 years. And there's probably long-term consequences that we haven't been paying as much attention to as we could.
Sorry, Nargis. Just, you're asking about the long-term, which particular complications?
So the report suggests that it's actually not much has been investigated when the autopsy samples have been taken and they find out that these patients have a bone marrow fibrosis.
Oh, bone marrow fibrosis.
Bone marrow fibrosis. So I'm just curious that, does that fibrosis have any impact on the hematopoiesis itself? Like how often the people have the chances of having a bone marrow fibrosis? I have not come across with many studies. Yeah, these are like 5 reports.
Okay, so that's interesting. So just, I mean, to answer Marcel's point, yes, there's actually a fair amount of literature now about post-TB mortality, which has increased. Post-TB loss of lung function, which is important, and 20 to 25%. Post-TB cardiovascular mortality, which is considerably increased as well. But bone marrow, I've not seen.
Okay.
Mayara's on the call, and she's, we're just delving into all this now, but I don't know whether Mayara, you want to comment. Mayara Bastos. And I know Erwan, which looks a lot like Eva Kaufman, has a question as well. But Mayara, have you seen anything? Mayara, are you still there?
Sorry, I'm here. No, I haven't seen anything on this. Yeah.
Bone marrow?
No, we don't. We did no, but we can look.
Yeah, might be interesting to look. Sure. Okay.
Yeah, that's a good point.
Okay. So, is that Eva Kaufman behind the mask? Anyway, there's someone who has a question there.
Do you know what's the impact of the wasting during TB infection and TB death? Like, have people tried to supplement a lot of nutrients to these patients, for example? Because, I mean, from the animal models, we know that to feed them can increase the pathology and can lead to a faster death.
Interesting.
But has somebody tried in humans to actually increase the food intake?
So nutritional supplements, both micronutrients and also just kind of total caloric intake, have all been tried. They, again, grosso modo, because they're randomized trials, they have not shown any difference in outcomes, so mortality. Now, whether there's a subgroup that did worse because of what you're saying and others did better, I don't know. There is a large randomized trial going on now in India, actually with Anurag Bhargava, who's a former trainee here, who's doing a large nutritional supplementation trial in household contacts to see if it prevents TB. And they're also giving nutritional supplementation to the active TB patients as well.
Okay.
So if you ask me that question, I can relay it, put you in contact with him, and maybe he can provide further info.
And do you know if because iron is very unspecifically taken up, so the content of the iron in this nutrition, do you know if you look specifically for that?
I'm sure that Anurag has looked at iron, but he's not giving iron supplements per se. But again, I'm happy to put you in touch because he's, in terms of nutritional impact on TB and so on, he's really, really one of the, and obviously now randomized trials, and he's doing sub-studies and so on. So I'm sure he'd be very happy to correspond with you if you had specific questions. I'd be happy
Sure.
to.
That's interesting.
But I have come across with a study, if I remember correctly, it's in South Africa. So the people who are taking a kind of a drink having a more iron content, they are more susceptible to TB.
Okay, I know there's a lot of excess iron. Yeah, it's particularly in the iron pots used for cooking, etc. I've not known that. Madhu, are you familiar with that at all?
I think Madhu is gone, Dick.
Oh, okay.
Sorry, Nargis, Marcella and I, we have another meeting, we have to go.
Yeah.
So I
Okay.
apologize. Thank you very much again. That was fantastic talk. Thank you, Dick.
Thank you.
Okay, great. Thank you, Nargis.
Thank you.
Thanks, Maz. Thanks, everybody.